Hi everyone, my name's Lily Bentley. I'm a PhD student at the British Antarctic Survey and the University of Cambridge, and today I'm going to talk about some of the research I've been doing as part of my PhD on the habitat preferences in sympatry and allopatry of light-mantled and sooty albatrosses. The key ecological idea that I'm going to explore in this talk is a common one, that niches of competitors in ecological communities are shaped by mutual coevolution, which is what allows multiple species to coexist. And this argument is frequently used to explain divergent resource use or divergence in other trait metrics when they appear like this, so two competitors with non-overlapping distributions of a particular trait. Joseph Connell claimed that while it was common to explain the presence of species with divergent resource utilization curves as the result of coevolution and competition, true coevolutionary divergence of competitors is actually much harder to demonstrate than at first it might seem. Connell argued that there are three criteria that need to be fulfilled before we can conclude that competition is what has driven coevolutionary specialization. And the first was that divergence has actually occurred, which is the one we'll focus on today. And then he also offered further criteria that competition needs to be demonstrably the mechanism, and then there needs to be a genetic basis for this divergence. But as I said, we're going to focus on the first criteria, which is identifying whether or not divergence has actually occurred at a species level. Because any study on one location or one population is kind of only one data point. So simply demonstrating that when species diverge living in sympatry, living together, is not sufficient to fully justify this argument. The divergence needs to persist when the species are in allopatry, or living separately from one another. So that then leaves us with two questions. Is habitat actually partitioned when the animals live in sympatry? And do the same habitat preferences persist when the animals live in allopatry? And in order to investigate these kinds of questions, what we need is a species pair, ideally reasonably closely related, that could be found living in sympatry and in allopatry. The Fabettria albatrosses offer us an excellent system through which to investigate this question. So the light-mantled albatross, here on the left, marked in light blue, Fabettria palpebrata, and the sooty albatross on the right, marked with dark blue, Fabettria fusca, are the only two species in their genus. They have similar morphology and breeding phenology, and they both have a circumpolar distribution. So if we want to understand the way in which this pair of species utilizes the habitat they have access to, first of all, we need to understand where that habitat is. In order to do that, we need to track them. And as many of you know, tracking large predators is both costly and very logistically challenging. So to ask this question, we needed to collaborate. And so at this point, I want to acknowledge the data collection work done by colleagues from 9 institutions in multiple countries. And thanks to their work, I now have compiled a data set with GPS and satellite tracks from 7 breeding islands, covering the years 2002 to 2018, with close to 150 individual birds. So what I'm going to show you now are tracks from the early breeding season, the incubation period. This is between November and January, and adults are sitting on eggs, or a couple are guarding very freshly hatched chicks, but the adults are foraging one at a time. There are also plenty of tracks from later in the breeding season to analyse, but because foraging behaviour can change quite a lot between breeding stage, I'm only going to focus on the tracks from incubation for the rest of this talk. So in this panel, we can see our tracks in allopatry. And in terms of sheer latitude, you can see that the light-mantled albatross in light blue breed in allopatry, so the only species there, at the southernmost islands in their range, in South Georgia and at Macquarie Island. Sooty albatrosses, in dark blue, breed in allopatry at the northernmost islands of their range, so Tristan, Gough and Amsterdam. So on this map, every island marked with a star has a single species of Fabettria albatross breeding there. On the right-hand side of this slide, we can see the islands where both species breed in sympatry. So both Marion and Crozet have both light-mantled and sooty albatrosses breeding there. And you can see a little bit of overlap right next to the breeding islands themselves, but by and large there's a very clear segregation where sooty albatrosses forage to the north and light-mantled albatrosses forage to the south. It's reasonably clear to see here that foraging habitat just isn't shared between these two species. But simply showing segregation of habitat use in sympatry doesn't demonstrate that this is an evolved specialization. For this preference for warm and cold foraging waters to be a true coevolutionary niche divergence, we'd need to see if these preferences persist in the absence of their competitor. So in order to do this, we need to know more about the oceanographic characteristics of each area that the species uses, as well as what they have access to. And it's important to consider access because the sooties breed in the northern 2/3 of the range, while the light-mantled albatross breed in the southern 2/3 of the range. In order to understand more about each part of the ocean that's favoured by each species, we can use satellite detected data. So here we have an example of a static file, so ocean bathymetry, how deep are the parts of the ocean that the bird travels over. And though that's usually a static value, we also have dynamic variables. So looking at spatially and temporally explicit data points for each latitude and longitude of the foraging track of the bird. And so here we looked at chlorophyll, sea surface temperature, mixed layer depth, current speeds, and sea level anomaly. And I'm going to focus on just the results from one of these variables today because I think it really accurately captures the pattern that we find. In order to understand habitat preferences, what we need to do is compare the used or the observed habitat with the available or the expected habitat. And what I'm going to show you some plots with a hypothetical image in shades of orange and a real track in black, and you can see that on the top left here. So if you look at the top right, you can see in order to sample all of the available habitat, all of the orange area underneath the geographic space, we retained the track shape to keep the physiological constraints the same for the bird, and then measured all of the shades of orange available to that animal if they moved off from the island in any direction with no specific intention. What we can then create are density plots of the expected type of habitat a bird would encounter if they don't have any preferences, and you can see that on the bottom right. And we can also create a density plot for the values of the observed habitat use, and you can see that on the bottom left. So in this example, you can see that the actual habitat used is much more clumped up at one end of the graph than the entirety of the habitat available. And that to us indicates some kind of preference, some kind of specialization, and we're not seeing habitat use driven just by random chance. So hopefully that example is clear. So now what I'm going to show you are the real values, just what we find for sea surface temperature. So here we can see a profile of the available habitat for all species in all conditions. So the top row shows light-mantled albatrosses, while the bottom row shows sooty albatrosses. The left-hand side of the plot shows each species in allopatry, so alone, while the right-hand side of the plot shows the species in sympatry, living together. The yellow bar is just used to highlight the peak of the distribution, and that peak happens at around the temperature of the nesting island because 100% of all of the samples start and end at the same nesting island, so that's why we see a spike of temperatures at that point, and then more variation the further away they go in each direction. And you can see that that peak of availability, that yellow line, is exactly the same for the birds living in sympatry because they're in the same place. Whereas the peak availability for sooty albatrosses in allopatry is warmer than it is for light-mantled albatross because sooty albatrosses nest at more northern islands. So when we overlay the actual habitat use of the light-mantled albatrosses, what you can see is that in both sympatry and allopatry, we've got a clear preference for cold water areas. And I've highlighted the same area in green in both graphs, and that's temperatures of less than 5 degrees Celsius. And as you can see, there's a really big bias for birds to target these areas. What's quite interesting that we see next is that when we overlay the sooty albatross habitat use, we do not see the same pattern in sympatry and in allopatry. So when living alone, sooty albatrosses target cold areas of less than 5 degrees Celsius. But when they're living in sympatry, sooty albatrosses avoid these areas. Instead, what we see is that they forage in warm water zones of 15 to 20 degrees. And if I highlight that same area on all the graphs, you can see it's an area that light-mantled albatrosses basically never access. And even though there's heaps of warm water really close to where the sooty albatrosses breed, instead they choose to travel much further to access colder foraging waters. So if we quickly return to Connell's ideas, we actually fall over at the second hurdle. We don't see this persistence of preferences when the species are in allopatry. So no true species level divergence in resource use has actually occurred. So in this case, the observed niche segregation is not the result of coevolution under competition. Rather, I think behavioural plasticity is the most likely explanation for why we observe such stark habitat segregation when these two species live in sympatry. And understanding why we see this pattern of habitat use in sympatry requires further investigation. possible explanations for the habitat segregation that we see in sympatry might be simply that sooty albatrosses are poorer competitors. They lose access to their preferred areas and have reduced success because of this. Alternatively, they might be a more adaptable species without fixed preferences. They can do equally well in less crowded warm water than they would in competitive cold water, maybe even better. In theory, there might be character displacement occurring in the sympatric populations of sooty albatrosses, somehow developing a warm water adaptation, but this seems unlikely given that if it was possible to adapt even further to warm water, you would expect to also see that at the populations living in much warmer water areas, and that's not what we see. We see a continued preference to forage in the cold. So the next step here is to work out if we can identify any quantitative difference in factors like egg weight, chick growth rate, or breeding success that might hint as to whether or not these warm water trips are more costly for sooty albatrosses breeding in sympatry, or if there's some kind of compensation occurring. I'm also working on repeating these analyses across different breeding stages because these sorts of birds often show quite different foraging behaviours at different stages in their breeding cycle. Ultimately, I think if we can better understand how marine predators develop habitat preferences and whether those preferences are fixed or flexible, it's going to give us a much better idea of the extent to which they're going to be able to cope with future climate change, and also how we can best manage them in terms of their risk of being bycatch in commercial fisheries. Given that the sooty albatross is endangered and the light mantle is also near threatened, there's a lot more to learn about these species, and understanding the ways in which they use the ocean is a really big part of that. Thanks very much for listening, and I hope to hear from you in the Q&A.